Don't steal bikes. Okay. Anyways, so we started off this unit talking about the cell cycle. So the cell cycle is like the life cycle of the cell. So we start with, well, let me back up. You can't just divide the cell without preparing to divide. Otherwise, you're going to have a cell with half the amount of DNA, half the amount of cytoplasm, half the amount of other cell stuff. So before we're going to divide the cell, we need to go through some different phases where we essentially grow the cell and make copies of everything. We break, we call that interphase, broadly speaking. An interphase has G1, S, and G2. G1 is, notice how we have unduplicated chromosomes, so we haven't made copies of the DNA yet, but the cell is just kind of doing some basic normal cell stuff. Maybe you're kind of growing some of the cytoplasm, making some copies of other organelles. S phase is S for synthesis. We are specifically synthesizing, making another copy of all the DNA. That's the important thing to understand with S phase. G2, we're going to be doing even more cell growth, but we're also like really making sure that everything is all set to go for cell division. Make sure we didn't mess up anything with the DNA here. We didn't duplicate the DNA, like we didn't do it like incorrectly. Then once everything is all ready to go, we have M phase, where M phase, the mitotic phase, that's going to be mitosis, and then you have cytokinesis, where you're going to split up the cytoplasm. Okay. This right here is showing something called G naught. This is representing cells that like don't divide. So that would be like your nerve, your muscle cells. Or even sometimes like these can be cells that like we found errors during while we're preparing for cell division. We'll put them in G naught and like label them for destruction. They call that apoptosis. Okay. My cursor over here. All right, so going through one things you want to be able to recognize, be able to do is describe and recognize a cell in these different phases of the cell cycle. So a cell that's in G2 of interphase, so the very end of interphase, right before it goes into cell division, what you'll notice, the key thing to picture is spaghetti. Think spaghetti, okay? Because before the cell goes into cell division, the DNA is uncondensed. It's uncondensed because we need to use the DNA. You don't want to condense down the DNA completely, otherwise you can't use that to do gene expression to make your proteins. So that's why I say spaghetti. It looks like a big bowl of spaghetti. There are these two centrosomes that are going to be like kind of, eventually they're going to migrate to opposite ends of the cell, but here you'll notice that they're specifically located on one end of the cell. That's where your mitotic spindle is going to come from. And that's really the main thing. I think noticing the chromosomes is the biggest thing to differentiate interphase from like specifically prophase, because both of them you have the nucleus intact. Eventually the nucleus breaks down to divide the DNA, but here nucleus is intact, but now the DNA has actually condensed into those chromosomes. Technically you will begin to see those centrosomes migrate to the opposite ends of the cell. It kind of depends on the picture you see. Maybe like some pictures, the centrosomes are closer to each other, so it can be a little trickier to tell if it's interphase or prophase. Okay. Yeah, you'll see the centromere. The centromere, that's where the sister chromatids end up like connecting to one another. So on each line here, this is a sister chromatid, and that's a centromere. That's a centromere. Whenever, before you did S phase, you only had one sister chromatid. Then we did S phase. That's where the second sister chromatid came from. They're identical copies of the DNA. Each has a centromere. And then they also are connected along their length by these things called cohesins. Cohesion, things are sticking together. So cohesins are these protein complexes that keep the sister chromatids together. Eventually we want to separate the sister chromatids. We'll have to break apart the cohesins. Anyways, so there you go. That's prophase. Now then there's pro-metaphase. So PP-mat is the mnemonic I gave you. So prophase, pro-metaphase. So in pro-metaphase, the nucleus has actually gone away. You can tell the difference between prophase, metaphase, the chromosomes have lined up in the center of the cell. So to tell the difference between those two, kind of extremes is pro-metaphase. The chromosomes are migrating towards the center of the cell. They're kind of on the journey towards the center of the cell. The mitotic spindle, the spindle fibers is more pronounced here. So you actually see those fibers pretty well. There's two different kinds of microtubules that you need to know. There's kinetochore microtubules and non-kinetochore microtubules. So once the cell is going to be, I don't know why I erased my sister chromatids. When the cell is going to like have to divide up those chromosomes, you have your centromere on each chromosome, but then we are going to end up building this other protein complex called the kinetochore around the centromere. The kinetochore is where the kinetochore microtubules will attach themselves. So here they drew it with like it's a little bit harder to see, but like that blacker looking color. That's representing the kinetochore, which I have in red right here. Okay, that's where those kinetochore microtubules attach, and those are going to be the ones that actually pull apart the sister chromatids. The non-kinetochore microtubules are just going to extend the cell. Because, we got to divide up the DNA, but we're also going to need to divide up the cytoplasm. So that's really kind of the role of the non-kinetochore microtubules, to extend that cell out. Okay. All right, then metaphase. Metaphase is arguably the easiest one to recognize because you actually can see the chromosomes along the middle of the cell. The metaphase plate is referring to the structure that where the chromosomes actually line up at. And then, notice your centrosomes are at the complete opposite ends of the cell. And there you go. Not a ton to say about metaphase. Anaphase, A for away. Metaphase, they meta in the middle of the cell. Anaphase, A for away. They're moving away from each other, or you can think of like the two A's are separated by the N. Like we're each A being a sister chromatid. So in anaphase, we end up pulling apart those sister chromatids, and they're moving to opposite ends. And then telophase, you begin to get your new nuclear envelope forming. So telophase and cytokinesis, they happen kind of simultaneously. That's why they're on the same slide. So as the nuclear envelope is forming, because mitosis, we're doing nuclear division. We're just dividing the nucleus. It's all about dividing up those chromosomes. Cytokinesis is dividing up the actual cytoplasm. That's why it's cytokinesis. Kinesis, movement, cytoplasm. And then the cleavage furrow, remember that's only in your animal cells. Animals have cleavage, plants don't, as far as I'm concerned. Plants will get a cell plate. I think I have a picture of that on a different slide. But there you go. That would be cell division, the different phases of it. So be able to recognize a cell that's like in that, or be able to describe a cell, as you saw like when we did at the FRQ practice, be able to describe a cell that's in those different phases. A little bit more details on like how we separate those sister chromatids. So if we look at this picture here, what's going on is whenever they were trying to figure out when the sister chromatids separate, where do the microtubules shorten at? Do they shorten at do they shorten from like here? Are they shortening at this end, or are they shortening at this end? So what they did was they radioactively labeled the entire spindle. That's why this is like fluorescing a yellow color that they could see. And then they used a laser to remove some of that fluorescence. So they made a mark right here. And the purpose of that is, if you can have a clear mark, you can then notice, well, which end will shorten, that end or that end. And as you see, the end to the right of the mark, the end that is closer to the kinetochore, that end is shortening, not the end that's closer to the centrosome. So what that means is when we're doing anaphase, the microtubule, that's what they're trying to show here with the tubulin subunits, the microtubule is shortening at the kinetochore end, the end that's attached to the sister chromatid, not the end that's at the centrosome. And that happens in animal cells. Some other different organisms will actually shorten at the centrosome end, but for our cells, we would shorten at the kinetochore end. Truthfully, for like the AP exam, I don't know how much they would really care about that, but it's kind of like more of like recent developments and like the study of how the sister chromatids get separated. So how the sister chromatids separate is two things. That's one of them, where they separate at the kinetochore end. The second is the motor proteins. So while the microtubules are being disassembled at that end, there's motor proteins that are attached at the kinetochore. So that's another function of the kinetochore, where the motor proteins can attach at. The motor proteins are like those things like no head, and they just like attach things. Well, they have a head, but they have like no arms. They just have like legs, like no torso. They like their heads like attached to the kinetochore, and then they're just like walking along the microtubule, pulling those sister chromatids apart from each other. Okay, so that's the second way that we're separating those sister chromatids. Okay. And then I think there's one more thing I need to say on this slide. It's not on here, but like just keep in mind, I mentioned that the sister chromatids are, like when I drew them, I said that there were cohesins holding them together. If we're going to do anaphase, we're going to have to break apart those cohesins. Just something to keep in mind. All right. As I mentioned, animals will do the cleavage furrow. Know that the cleavage furrow is a contractile ring made of actin. That's an important detail to know. It is an actin ring. We went over like the three parts of the cytoskeleton, like actin, microtubule, microfilament. The spindle fiber is made of microtubules. The cleavage furrow is made of actin. Okay, that's kind of a detail that the AP exam likes to have you know. And then if we look at the cell plate that plant cells will form, what happens is you would get vesicles that come from the Golgi apparatus that would have the materials you would need to build that new cell wall. So know that the cell plate, reason why plant cells have a cell plate is that's going to become the new cell wall. Okay. Any questions so far? Doing okay? All right, looking at some regulation of the cell cycle. So they did an experiment, and they were like, all right, if we took a cell that was in S phase and a cell in G1, the question was, where are the signals that tell the cell when to go through the cell cycle? Where are they located? So what they did was they suspected maybe it's in the cytoplasm. So how they tested that was if we take two cells that are in different phases of the cell cycle, right? Notice how there's twice the amount of DNA in S phase than G1. We combine, if we fuse the cytoplasms, what will happen? And what they saw was if you fuse the cytoplasm of these two cells, whichever cell is further along in mitosis, that cell will send its signaling molecules, the one that's further behind, and tell that one to move to meet where that cell, the cell that's further along, to meet it where it's at. I don't know if I said that properly, but this one was in G1. It immediately goes to S. But notice how it didn't actually complete. Well, here it duplicated the chromosomes, but like it could, like if you look at the second example, this cell that was in M phase, this cell that was in G1, it sent that cell all the way to M phase without going through S phase. That's kind of an important thing. So even if like, in other words, it'll skip through different cell cycle controls, which I'll mention in a couple slides. The cells will immediately go again to like the latest phase of like where that cytoplasm's at. So the point of this is the cytoplasm controls, there's molecules in the cytoplasm controlling which phase of the cell cycle to be in. And that's where we get into these checkpoints. So there are a few different checkpoints to know. The first one is the G1 checkpoint. G1 is the first phase of interphase, right? So before we go from G1 to S phase, we're going to take a look at the cell and be like, all right, we need some sort of go-ahead to make sure that cell's done some sufficient growing, or maybe we're doing some, the cell is trying to sense like the environment around it and be like, all right, do we need to divide, right? We'll talk about density-dependent inhibition, the Anchorage dependent inhibition. Do we actually need to go through cell division? If we don't, and then G0 would be, all right, we're going to just do a non-dividing state. We don't need to go through cell division right now. Then, okay, we need to do it, then we would go through it. The M checkpoint is a super important one. That one is where we're going to like actually make sure the chromosomes are lined up completely at the metaphase plate. Because think about it, if the chromosomes aren't lined up on the metaphase plate properly, what can happen is you don't get the right number of chromosomes going to each of the two new cells. That's the important part of the M checkpoint. The G2 checkpoint What do you think we do in the G2 checkpoint? They don't really talk about it specifically in this slide. M checkpoint is checking that the where all the chromosomes are aligned on the metaphase plate. G2, what would G2 be checking? What came before G2 that you would need to check? Thomas? Yeah, because before G2 was S phase, where we synthesized the DNA, we got to make sure that we made a copy of all the DNA properly. Okay, make sure there's no errors. Because if you don't go through the G2 checkpoint properly, you could end up dividing a cell that maybe has some mutations that might lead to cancer. Okay. All right. Speaking of like these checkpoints, the question is, well, what kind of molecules are signaling that we need to go from this phase of the cell cycle to the next phase? And the main kind of molecule I want you to know is the cyclins. There's a bunch of different kinds of cyclins. Just know cyclins. Know that like to go like within G1, there are G1 cyclins, there are M cyclins, there are G2, there are S cyclins, and those will peak at the time. Like we need to go through like G2, the G2 cyclin will peak. When you go through the M phase, the M cyclin will peak. And I think I have a picture, yeah. So I have a graph here on the next slide that would show those peaking. Anyways, the other thing on this slide I want you to know is what's called the cyclin CDK complex. So cyclin is the protein that's doing the signaling, telling the cell to do these things. CDK stands for cyclin dependent kinase. You should be able to break down that word at this point. Cyclin, that's the substrate. This is an enzyme. The substrate is the thing the enzyme works on. Dependent, it's a kinase that depends on cyclin binding. And kinases do what? You know that. Told you kind. And if you like don't think about it too much, the D in kind looks like a what? A P, yeah. Looks like a P. So kinases, they attach phosphate groups. Now that we're all on the same page. Kinases attach phosphate. So the cyclin-dependent kinase, CDK, attaches phosphates to cyclin, and that would then make it activated. Okay? All right, so this is what I was talking about, where the different kinds of cyclins peak at different phases of the cell cycle. So if we're in G1, the G1 cyclin's a little bit kind of an exception to this general rule, where it will actually technically peak in the middle. You don't even know where these peak at, just the idea that like there's a G1/S cyclin that peaks between G1 and S, right? The S cyclin peaks slightly after the S phase, things like that, things of that nature. Okay. Yeah. All right. Getting into regulation of the cell cycle. So what they want to figure out is like how do we signal cells to grow? So the point of this slide is to look at something called platelet-derived growth factor, PDGF. And what they saw was if you took a cell and you try to grow that cell on culture, so like culture is like basically this like substance that has nutrients that can feed cells to allow them to grow. If you grow them on that culture that doesn't have PDGF, you don't really get much cell growth. But if you grow them on the culture that does have the PDGF, the cells grow. Kind of a simple idea, but that's what they were trying to test there. Okay? Well then, there are two kind of like ways cells will sense, do we need to divide or not divide. Here they're looking at something, two different things. One is called Anchorage Dependence, and the second is Density-Dependent Inhibition. And the names of it kind of give away what they do. Anchorage Dependence says that cells are only going to divide, or should only divide, when they're anchored to something. That something is usually the extracellular matrix. Because think about it. Cells will like organize, cells of the same type organize into something called tissues. Those tissues then make up organs. So in other words, like you need all the cells to be in their proper positioning. Think of like an orchestra or a sports team. Everybody's got to be coordinated at the same time. So they want to be anchored to the extra. It's like you know, if you're like doing a play and they might have like markers on like the stage floor telling people where to go. That's kind of the idea of anchorage dependence. Cells will sense the extracellular matrix and anchor themselves and they grow. Density dependent inhibition is densities referring to like, you know, how many cells in a certain volume, cells should only grow when they sense that there's an opening around them. And that's what they're showing here. So if you had imagine this is like a cut, the cells around that cut or that removal will sense that there's a space to grow, and they'll grow to fill that opening. Okay? The main point of going over this is cancer will happen. A lot of things can cause cancer, but usually some sort of breakdown in density-dependent inhibition or anchorage dependence would lead to cancer. So in a cancer, that's cells that divide without, they just keep dividing uncontrollably. They don't sense the signals that tell us, hey, you need to stop dividing. So they divide beyond a single layer here, so they're violating the density-dependent inhibition. And a lot of them won't show anchorage dependence either, meaning they don't have to be anchored to the extracellular matrix to keep dividing. And why is that why is that such a big deal? That isn't maybe completely obvious. Spreading, yeah. So cancer cells that lose anchorage dependence, they lose that ability. Those are the ones that can metastasize and become the malignant tumors, the ones that are really, really serious. If you have a cancer cell that lost density-dependent inhibition, that might lead to a benign tumor where it's still a tumor, still a growth, but at least it's kind of localized, maybe here, and we can do surgery and remove it. But when they lose anchorage dependence, that's when they can move to other areas of the body. All right. Another thing that cancer cells not lose, but I mentioned how for cells to grow, they need a growth factor, right? Cancer cells don't need growth factors to grow. So they're not responding to the signals that would normally tell them to stop. There's kind of a few different ways this could work. They could either make their own growth factor to stimulate themselves, or they could just grow without the growth factor. They don't need that growth factor binding to the receptors in the cell membrane to grow. It just does it normally. Or they have abnormal cell cycle control systems. So I mentioned how we have certain checkpoints as you go through the cell cycle to make sure we divide at the right time and everything's all ready to go. Cancer cells, that cell cycle control system can break down. They're no longer they didn't duplicate the DNA correctly, and they don't care. They're going to divide anyways, which can lead to some other problems with breaking down certain losing control of certain genes that we might need to control the cell cycle. What can happen with this and with a lot of these is mutations. Basically, you get mutations in they've identified certain genes that they would call cancer causing genes, or if you have mutations in these certain genes, you're more likely to develop cancer. Those genes would be genes that are especially important in controlling the cell cycle control system and controlling the anchorage dependence and density dependent inhibition. So genes that are involved in all of that, those are the ones that are especially serious if you get mutations in those. Okay? If you get a mutation in a gene that's controlling lysosome, I don't know, some other thing that's important in its own right, but maybe it's just the cell isn't as efficient, that won't necessarily lead to cancer. Not all mutations lead to cancer. That's what I'm trying to say. Cells that have undergone that process where they kind of lose the ability to respond, the breakdown in the growth factoring signaling and then the anchorage and density inhibition, we say those cells have been transformed. They've undergone transformation. It's kind of a random word, but just something to know. No benign versus malignant versus metastasis. Benign tumor, that's where it's just localized. It stays in one spot. That's where I was saying the tumor still has the anchorage dependence, where it's not like it doesn't have the ability to travel. But then malignant, they've lost the malignant think bad. Malignant, they can actually travel. They can travel and conquer other parts of the body. And when tumors have done that, we say it's metastasized, or the process is metastasis. Okay. Looking at kind of walking through an example of how a tumor could go from benign to malignant. So if you're looking at the breast tissue here, so here's a tumor growing. Now, in order for this cancer to invade neighboring tissue, you're going to need some way to transport it. So think about it. Like for a tumor to spread, it needs some way to transport, and usually that's in the blood vessel or the lymph vessel. So what can happen is this cancer cell will lose anchorage dependence, as I mentioned a few different times. But the thing I want you to key in on here is they can then, the cancer cells can send out signals. They can send out growth factors, I'll just label it GF, and signal to the blood vessels and lymph vessels to grow towards them. It's kind of wild. The cancer cells can send growth factors to blood vessels and lymph vessels and tell them, grow towards me, so that I can then move through your blood vessel or your lymph vessel and spread to other parts of the body. It's kind of a wild idea. Okay, so they got to lose anchorage, and they have to have a blood vessel and a lymph vessel nearby. Okay, and both things have to happen. Like my sister got like skin cancer, and it was technically malignant. Like it had lost like the anchorage dependence, but the blood vessels and lymph vessels hadn't grown towards it, so they were able to like get it just in time, fortunately. But yeah, so with cancer, there can be a lot going on. It's not like any one thing. You kind of need a lot of different factors for it to get to the stage where it travels all the way through your body. Okay. All right, looking at some cancer treatment. If it's a localized tumor, so it's something that's more benign, hasn't traveled too far, you could just do some radiation and just kind of, or even surgery, and just kind of remove that tumor from that area. But if it's been like, if it's spread, if it's metastasized, then you have to do chemotherapy. And I want you to know how chemotherapy works. So chemotherapy drugs, they typically they target cells that divide frequently. Because think about it, cancer cells divide frequently. That is essentially their issue. The problem with that is a lot of the side effects from cancer, because chemotherapy targets cells that divide frequently, things like your skin cells, epithelial cells, your kind of skin cells. The same kind of cells that make up your skin, they also make up your digestive tract. That's why people who are on chemotherapy, they have like digestive issues, because that digestive lining is being kind of interfered with. That's why they lose hair, because the cells that kind of make up your hair follicles, they divide very frequently. Immune issues, your immune cells. So immunocompromised, your immune cells divide very frequently. So it's definitely a trade-off, right? Where we're kind of making the trade-off, trying to save your life, but in the process, like, you're kind of really affecting some of these. You're kind of creating a lot of these pretty severe side effects. Last thing on the cancer thing, and then we'll take a little break, is kind of the future of cancer treatment. Is how can we, like, if we understand the actual changes in the DNA that lead to cancer, maybe we can better personalize the chemotherapy, or try to come up with some other treatment that reduces these side effects. Or you find that right kind of combination of cancer drugs that would work better for like breast cancer versus colon cancer. Or somebody within, even like if you're treating colon cancer, somebody who is like specifically, maybe even of this race, maybe they can identify, or trying to identify certain genes that like people that are like of a certain race or ethnicity tend to have that specific kind of mutation in their DNA, that kind of specific copy of that gene. Okay. All right, let's take a little break, and then we'll do the rest of it.